Hello. Hi, everyone. Welcome to this month's VanBug. We have 2 more of these this year, March and April, I think. We have 2 more talks in March and April, right? Yes. So look forward to joining us there. But for now, we'll have some announcements, and then we'll have an introductory talk, or featured talk. And then after, there will be pizza and refreshments in the room over there. I'm just going to, here, our sponsors at VanBug are Ecoscope, the Graduate Studies Program in Bioinformatics, the BC CDC Foundation for Public Health, the Canadian Bioinformatics Workshop, the Pacific Institute for the Mathematical Sciences, GeomeBC, Adversarial, BC Cancer, and we are currently webcasting this event, which will be available later on YouTube as well as live now. So thanks to everyone on the organizing committee. And these are our social media accounts and contact information if you need to contact us. I have an announcement from HackSeek, and we're going to be joined by someone who's organizing HackSeek. So here we are.
Oh, thank you. Thank you. Hello, everyone. My name's Artem. I've been around for kind of too many years at this point. But I just want to tell you about an event that I'm helping co-organize. It's called HackSeek. If you've never heard about HackSeek before, this is a 3-day genomics hackathon. So you form groups of, say, between 4 to 8 people. Someone as a team leader comes, proposes a project, and then you work on that project to create something, usually software or a pipeline, over that 3 days. And then, we have a winner and a lot of networking and stuff. This year is really special. We've teamed up with the RNA Society. So this is the biggest RNA meeting in the world, and it's happening in Vancouver in May. And so all the world's leading RNA computational biologists are going to descend on Vancouver. And this is our chance as kind of the Vancouver community to shine, show them what we can do, and also learn from them. So if you're interested, participant applications are going to be open for probably the next couple of months, but we're really looking for team leaders, and it would be awesome to have some local kind of Vancouver-based team leads that will be able to recruit people internationally. So check out the website, it's hackseek.com. Thank you.
Thanks, Artem. So up first, we have our introductory speaker. We were pleased to welcome Hossein Sharifi Nagavi, a PhD student in Martin Astor's lab, and he'll be talking to us about transfer learning and machine learning in pharmacogenomics. Hossein.
Awesome. Thank you so much for this opportunity. Good evening, and thank you for the introduction. Today I'm going to talk about transfer learning and drug response prediction. Let me first thank you, people who were involved in this study. Shuman Pink, our brilliant undergraduate student, and Olga Zolotareva, our visiting student from Bielefeld University. Imagine you have a data set of very, very nice drawings. And your goal is to train a machine learning model, for example, deep neural network, to classify them as accurate as possible. The problem is that you don't have enough samples. So, and we're all grad students probably. We don't have money to recruit professional artists to draw images for us. So probably would step up and draw them yourself. But if you're a fake artist like me, this is not quite what you are looking for. You can also use internet and look up online for finding more images. And it's good. You'll get enough samples. But the problem is that if you use the samples that you found online to train your model and test it on the data that you care about, those nice drawings, in reality, instead of targeting your goal, you're off target a lot. Because your train and test data are coming from different distributions. As you see in this, for example, image, the nice drawings are very, very different than those you drew or those that you found online. Transfer learning attempts to solve this problem. In traditional machine learning, you have separate learning systems for each goal and each task that you want to study. But in transfer learning, you have one learning system, and you want to use the target task, the task that you care about, in our case, the nice drawings, and all the other relevant data that you could find. We call them source tasks or source domain. For example, those that we found online. You want to use all of them together to eventually achieve a better performance to the task that you care about. Transfer learning can be categorized into three categories: unsupervised, transductive, and inductive. Unsupervised, as the name indicates, you don't have any label information in source or target. In transductive, also known as domain adaptation, you have only label in your source the data that you found, the big data that is related to your goal but is not your goal, but you don't have the label in your target. And the tasks are the same, meaning that if you have 10 classes to predict in the source, you have the same 10 classes in the target. Inductive transfer learning is a little bit more tricky because you have label in both source and target, but your tasks are different, your domains are different, so it's a harder scenario. Today, I want to talk about drug response prediction, challenges that we have in drug response prediction, why I think transfer learning is a solution for that, and the method that we developed. For drug response prediction, ideally you have a group of patients, you get some omics data types from them, for example, gene expression. You screen them in one or two drugs, build your computational model, and try to predict drug response for future patients. But patient data with drug response is very limited and or not publicly available because of the privacy issues. And you cannot treat a patient with hundreds of drugs independently or even in combination just to study the effects. It's impossible. Alternative solution is preclinical resources like cancer cell lines or PDXs. Very similar idea. You get some omics data type. Now you can screen them with more drugs separately or in combination. You build a model and hope that model generalizes to patients. We have many, many discrepancies in drug response prediction. For example, gene expression data obtained from cell lines and patients obviously are very different. A bunch of cancer cells on a plastic dish are very, very different than human body. On the other hand, measures of drug response is also very different between cell lines and patients, because in cell lines you have, for example, IC50, the concentration that kills half of the cell. But in patients, you cannot increase the concentration step by step. It's impossible. So you should use alternative measures, for example, changing the size of tumor as a proxy for the response. Even this study in Nature shows that two well-known cell line data sets, they also disagree and have their own inconsistencies. So with all these problems, you have two options. First, you can train your model on cell lines and validate it on cell lines, stay to the preclinical data. You will lose translation because you don't know if your model is going to work on patients. But on the other hand, you minimize the risk of facing discrepancies between your train and test data. Your second option is that you train on cell lines and validate it on patients, clinical data. Now you can study the translation of your model, but on the other hand, the risk of discrepancy is maximal. But ultimately, because patients are those that we care about, we want to see how good our model is for patients. You have two ways to address these discrepancies. The first way is that try to obtain data in a more standard way. For example, a very good example is PharmacoDB resource at U of T, that they gather all the preclinical data and try to pre-process them and analyze them in a uniform way so that you can reliably build computational models for them. Your other option is that you design computational tools that can handle these discrepancies. For example, this method, Precise, they design a method based on principal component analysis that takes the gene expression from cell lines and patients and adapt them. But as I said, drug response prediction is unique because not only you have input space discrepancy, gene expression, difference between gene expression of cell lines and patients, you also have output space discrepancy, different measures of drug response between cell lines and patients. Because in cell lines, it's a continuous measure of drug response, for example, IC50 or area above dose response curve. But in patients, it's a categorical measure. Based on what I introduced at the beginning, this falls into the hard scenario, inductive transfer learning. And there is no method, computational method, that can address these discrepancies at the same time. So we decided to design one. This slide seems a little bit complicated, I agree. I'll try to walk you through it. Our method takes gene expression from cell lines, the source data, and gene expression from target data, the patient. And in the first component, a feature extractor, you can think of it as a very, very complicated PCA. Extract features from these different data and map them to a feature space in lower dimension. Now, remember my two goals. I want to make this output space discrepancy between cell lines and patients, and I want to make the input space discrepancy solve, different gene expression between cell lines and patients. For the first problem, we design a multitask subnetwork. This multitask subnetwork takes the extracted features and makes predictions simultaneously for both cell lines and patients. It has a regression component for cell lines and a classification component for patients. Remember that I told you, in cell lines, the drug response prediction is a continuous problem because of the measure, and in patients, it's a classification problem because it's, for example, based on changing the size of tumor. Jointly training cell lines and patients to make predictions helps also the limited sample size that we always have for patient data, for example. And moreover, I can use this multitask subnetworks to assign cross-domain labels because I only have continuous label in cell lines and categorical labels for patients. But when I train them together, I can use the classification component to assign binary labels to cell lines, or I can use the regression component to assign continuous label to patients. So in this way, I can solve the output space discrepancy. Now I have the same labels for both cell lines and patients based on the problem that I'm studying. But I still need to solve the input space problem. For that, we use the idea of adversarial learning. In adversarial learning, you have two competing components. In this case, my feature extractor is competing with these discriminators that I have on the right side. The feature extractor wants to extract features such that cell lines and patients look similar to each other. Imagine if in some space, this is my cell line and this is my patient, I want to map them to a space that they're here. Instead of being here, I want them to be here, closer to each other in a feature space. On the other hand, the discriminator wants to also be really good at capturing who is cell line and who is patient. So in a way, discriminators, they want to maximize their performance in catching which source this data is coming from. Is it a cell line or patient? And a feature extractor wants to minimize its error of being captured. It wants to learn a domain invariant representation such that these discriminators, they have no idea whether this sample is coming from cell line or patient. And this is the idea that in computer vision people are using in the context of generative adversarial networks. So by using these discriminators in the global level, which ignores the class label, and in the class-wise level, which considers if these two samples are responder or non-responders, it's a stronger constraint. By using these, I can be sure that these features are domain invariant. And by using the multitask subnetwork, I can be sure that I have the same labels for both cell lines and patients. To validate this method, we use different resources. All the gene expression data are obtained before treatment, and obviously the response outcome was after treatment. For cell line data, I use GDSC repository, a panel of 1,000 cell lines screened with different drugs. For patient data, we gathered data from TCGA patients with drug response in their record, different clinical trials, a PDX data set, and based on availability of number of patient data that we could find, we focus on those 4 drugs that you see on the other side. When I apply this data to my method, here is this table, which looks scary, but again, I'll try to walk you through it. The first 2 methods, they don't have any adaptation. They were purely trained on cell lines and validated on patients. The next 3 methods, they can only do input adaptation, meaning that they take the gene expression data and try to adapt them. And the last method is only can adapt the output space. As you see, our method in the last row outperforms all of them in terms of area on the raw curve, which shows that addressing the discrepancy in both the input and output side is more beneficial than not answering it, but thinking about it, not considering it, or only focusing on the input or the output. We also did an evaluation study, removed different components of our own method to study the impact of each part of it. We obtained a similar result for precision recall, and as you see, our method in its complete version in green outperformed the baselines and its own variants. So in summary, we propose the first method that can adapt both input and output space in the context of drug response prediction. We showed that this method can outperform a state-of-the-art transfer learning and state-of-the-art pharmacogenomics method. For more details, please read our drafts available on bioRxiv, and thank you. Questions, please.
If there are no questions, then thank you so much. So today, our featured speaker is Matt Pennell. He's an assistant professor at UBC in the Department of Zoology. He is I'm going to get this right, a Canada Research Chair in Biodiversity Theory and Informatics. And his background is in macroevolution. And he's focusing on building phylogenies and using them to test evolutionary hypotheses. So let's welcome Matt to the screen here. Oh, which one is it? Ah, good.
And do I just slide advance on the buttons? Yeah. Okay. All right. Okay, let me just stand here. Perfect. Thank you so much for inviting me here. It's a total privilege and totally terrifying to speak in front of an audience who is way more sophisticated than I am. I'm sure I'm going to look like a total noob that I don't know what I'm doing, but that's okay. That's why I'm here. So as Evan said, I'm an assistant professor at University of British Columbia, and I'm in the zoology department, which is basically the biology department. And I'm going to tell you today about a problem that's been bugging me for a long time, and I basically titled my talk, Learning About Evolutionary Dynamics from Molecular Phylogenies, but the take-home of my talk is something regarding how to avoid spending a decade on a problem that was unsolvable. So I'm going to work you through a problem in my field and talk about all of the attempts, including mine, to basically get at it. Then some way of thinking about whether the problem was solvable at all. And I think the answer is in my slide. And so you may not know this field very well. I'll try and explain it the best I can. Please ask me questions afterwards if that's what you want. And but hopefully basically the lesson of how I approach this problem might be actually interesting to you, and you can save yourself time, money, and tears. All right. So my basic background in this field called macroevolution. Maybe some people don't even know it. And basically, broadly speaking, it's the study of evolution above the species level. We're talking about diversification of life on Earth over long time periods. As beautifully depicted here by this paper that just came out in Science about the marine invertebrates through the Phanerozoic. And this kind of study about why life has been more diverse at different times in the past, and maybe what the drivers of that diversity are, was long the domain of paleontologists. People were working in museums, looking at rocks, categorizing species, and developing kind of mathematical models about why we might see changes in diversity at different times. And a big question since basically the 1940s is to ask, how have the rates of speciation, in other words, splitting, and extinction, obviously things going extinct, varied through time and across groups? What is driving different patterns of splitting and extinction? What's the role of climatic variables? What's the role of competition? These type of things. And this has been something that they've been doing in the fossil record since the 1940s. And there's lots of theories about factors that should affect these rates that I won't go into at all today. And then back in the 1970s, there was this huge revolution in the paleontology literature. And this is, I call them old school bioinformaticians. And these two dudes, along with a bunch of other people, were developing some really sophisticated mathematical tools for trying to figure out how to model changes in species number through time. This work is awesome, and it's still going on today, but there's some major kind of limitations and problems with taking kind of a fossil-based approach. And that's that most of our theories of biodiversity are about species, but it's really darn hard to limit species from the fossil record. Oftentimes you're looking at morphological types. It's hard to discriminate what you're looking at. Oftentimes our theories about why diversity has changed through time and across groups has something to do with the traits. Many of the traits we might think are important for changing diversity, such as molecular traits, are actually impossible to measure in fossil groups. And for a lot of groups, especially ones that are really interesting, sampling is really uneven and really sparse. Bacteria, for instance, we basically know hardly anything about the fossil records. We have no idea about what could have led to changes in bacterial diversity throughout evolutionary history. So this is basically not where I come in at all, but the core problem I'm interested in. So back in the early 90s, people were starting to build phylogenetic trees, looking at the evolutionary history of species from molecular sequences. This is back in the day when a tree was large. If you had a couple dozen nucleotides and maybe 10 species, which is obviously shockingly small today. But at this time, some people had this really great insight that perhaps you can gain a foothold on the problem of what caused variation in speciation and extinction rates using these kind of molecular data. And the idea is pretty simple, but I'm a pointer, but I'll just point my finger, and you can imagine, is basically we start off on the top hand there, we have some little diverging phylogeny, and over time it accumulates more species, but some species go extinct. And then at the end, we sample the process, but as we only have data from extant organisms that we can sample their DNA from, we're going to be missing a lot of species, all these things which don't touch the end. And so our reconstructed phylogeny on the bottom here is built from the genetic sequences of only the extant critters, whatever they may be. But the genius of these kind of early researchers was actually there is information in the branching structure of the phylogenetic tree itself about the underlying process. And if we can, for instance, assume that the rates of speciation and extinction have been constant throughout time, and we'll get to that, back to that assumption later. And then in the deterministic limit, the number of species in the group will accumulate according to the right-hand curve. And what's really remarkable is that even though we aren't actually we can't see any extinction events, all we have is the present-day species, we can actually get information on the two rates. And this happens basically because if you look at the far right-hand side, all the things around today haven't gone extinct yet by the nature of them being sampled today. So we have like a censored process. And we can leverage this kind of censored process to disentangle these kind of the splitting rate from the extinction rate. So even though we have only molecular sequences from a group of living organisms, we can kind of, given some model assumptions, reconstruct possible parameters of diversification. And just to kind of drive this point home, that here's 3 different scenarios on the top here of what our tree could look like. And each of these implies a different accumulation of lineages through time on the bottom. And each of these patterns of lineage accumulation perhaps correspond to a different evolutionary theory. We can estimate these rates and ask all sorts of interesting questions based on what we infer. And I told you at the beginning that basically this whole operation started on when we had maybe a dozen species in our tree of life. Now, a bunch of people, including in a very limited extent me, have been basically reconstructing the tree of life. And we now have trees containing tens of thousands of species for many groups of organisms. Jeff Joy, who's here in the audience, was a major contributor to the bird tree here, and I believe the sharks as well. So you can ask him about how they're made. And so we have incredible resources, and this is fantastic. And the hope and idea was that we could kind of apply this theory we were talking about earlier to understand what could have caused different groups to have more diversity than others. So the simple kind of model I described at the beginning is called the birth-death process. We're going to assume at the beginning that rates are constant through time in both splitting and going extinct. But just as the set of sequences we've had to work with have increased a lot over the past 2 decades, so has the availability of models. Now we have statistical models which have heterogeneity in the process in many different sources. And this is, of course, important because we think that these rates of splitting and extinction, which ultimately determine changes in biodiversity, could vary due to lots of reasons. You can imagine they vary through time continuously. They vary through time discretely. You can imagine a mass extinction event killing lots of things. They vary by taxonomic group. Some groups may intrinsically be splitting faster. It may vary as a function of traits, what features they have, maybe geography or whatever. The environment they're living in, there's lots of hypotheses about, say, why temperature may influence diversification. And there's also a lot of hypotheses about how competition among different lineages may, in fact, structure the patterns of diversity. And this is kind of captured with these other models. So basically, we can, in all of these cases, we can describe a stochastic model which has the features we're interested in. And we can write down a likelihood function which says how probable is our given branching pattern given some hypothesized idea. And we can test and compare alternative hypotheses. And we can fit models, estimate parameters, this sort of thing. And this has been awesome. A project I was recently involved in, we built a phylogeny of 448,000 bacterial species. They're not really species, they're called OTUs, whatever you want to call them. And we actually estimated rates of splitting and extinction from all these bacteria. And our primary discovery was basically these are very similar in lots of ways to birds and say, vascular plants and the rates at which they go extinct. And so it goes the way they speciate. And that this kind of demonstrated that bacteria are, in fact, diversifying through time. And even though we have basically no fossil record of these guys, we can kind of trace their average rates across time and compare them to different groups. Okay, so that's pretty cool. And I talked to you about this kind of came out of the macroevolution world, which I'm from. But this problem with slight modifications has been applied to other problems, which are super interesting. Just like the diversification of viruses during epidemics. These are a bunch of flus. And as you can see, they have different branching structures. And we can basically use the information on the branching structure of these viral phylogenies to, in theory, infer about things about how the epidemic's progressing. We can do things like estimate the R0, which is the basic reproductive number of viruses, using the branching rates on our molecular phylogeny. And we can look at things like, say, cumulative cases and stuff like this, and kind of get a real-time view of how this virus is diversifying. And we can ask whether certain, say, lineages of this virus are diversifying faster. Maybe they're more infectious than other strains for whatever reason. But it's all using the same kind of logic that I laid at the beginning. And we can also do things nowadays with cancer cells. So there's people doing some really amazing things, looking at the diversification of cancer cells within a human body, sequencing single cells at different parts of the body, and basically tracking the phylogenetic history of these groups, and looking at which different cells are splitting at different rates, and can we infer that, and basically use that to guide some kind of therapy in the future. So this is kind of the background of what we're trying to do and why we're trying to do it in the macroevolutionary context, in the viral context, and then in the cancer context. We can also think of other problems too. People have used these methods to study the evolution of culture. People have used these methods to study the evolution of B cells within a human body, these kind of things. And so these kind of splitting and extinction rates are super important for lots of different processes, and understanding them is a key problem in several fields. And today I'm going to try and pick this notion apart and try and get something that has been really bothering me for, like I said, a decade, is we have all these methods which have together been applied to thousands upon thousands of studies, and we've estimated rates for many groups for many time periods. We've tested many hypotheses. But the question that stands is, how confident can we be that we're getting to the truth, that the rates we estimate are real rates? And like I said, it's a strange problem because we're not seeing the whole picture. We're seeing a censored view. Whether we look at phylogenies of extant-only taxa, we're only seeing the ones that survive to the present day. Or if we're looking at viruses, for instance, we'll only look at the ones the only lineages we've sampled. And so we're always looking at an incomplete view of history and trying to reconstruct the rest. And so it's worth asking at a deep level, how confident can we be that we're actually getting somewhere, that our answers are converging on the truth? And so we have reason to worry that we might not be getting there, right? So some simulation studies, this is an old one from 2010, but there's a number of other ones that basically show, well, in these lines here, the little arrows correspond to different levels of what they call extinction fraction, which is just the rate of going extinct divided by the rate of splitting, just a single parameter. And basically, it says what the true value is, but if you simulate under different amounts of heterogeneity along the y-axis, sometimes you get really, really bad responses. In fact, when you have a ton of rate heterogeneity, the bottom case, you tend to converge the relative extinction to be either 0 or 1. And this is really bad behavior, no matter what the actual process was that you simulated under. So there's been this and a bunch of other studies which have kind of said maybe if our assumptions aren't valid, we might be making mistakes. But it's really unsatisfying to know, as you may experience in your own work, on a case-by-case basis, you can show that some method fails, but it's really hard to develop general theories as to when and where it should fail. And so that's what I'm trying to come up with here. And so the rest of the talk is basically going to be a mathematical derivation, and I apologize, but that's how it goes. So this is my collaborator, Stylianos Luka, who's now an assistant professor at the University of Oregon. He was a PhD student and a postdoc in the math department at UBC. And so what we were trying to do is basically come up with like a general theory that would tell us when and where such model fitting might succeed or might fail, and how confident we could be in the general results. So I'm going to walk you through this derivation slowly. And forgive me if it's not clear. I tried to do my best, but not the best presenter, of course. So consider an arbitrary model where you have two parameters, the speciation parameter, the rate at which these kind of lineages split, and the extinction parameter, mu, which is the rate at which they go extinct. We're going to say they can be arbitrarily variable through time, but constant across the tree, such that basically our entire tree has been generated by the same process. And these can be arbitrary functions. On the top part, we have the speciation rate is a linear function of time. Maybe as towards the present, rates have sped up. This may be due to changes in the environment or whatever. It doesn't matter. We basically have an arbitrary function. And we're going to assume arbitrarily that our extinction rates are constant. And so we can make any kind of complicated function you want and put it in here. But we'll just basically let these functions vary arbitrarily. So what's really remarkable mathematically is that, and this is something that's been known for a while, but not appreciated by many people, is for a reconstructed phylogeny, where you basically taking samples you have, the only information regarding lambda and mu, the splitting and the extinction rate, are recorded in a series of branching times. And we can make a thing called a lineage through time plot. So what this means is it turns out if you can take any tree, you can always convert it to a series of waiting times to the next branching event, depicted below here. Okay? And it turns out the actual topology of the tree is irrelevant for estimating diversification rates. It doesn't come into the picture at all. We can just decompose this single tree into this kind of series of branching times, and that gives us all of our information. So we can write down the likelihood, which describes the probability of our data given some hypothesis, in this case, the probability of our series of branching times given some hypothesized evolutionary model. And we can completely ignore the tree structure. And so this is really interesting, theoretically. Because what this means is if we take this tree here of, I can't remember how many fish there are here, but there's a lot, many tens of thousands. And this produces this beautiful graph. If we want to look at kind of if we assume, for instance, that the rates of diversification have been constant throughout the tree, which they haven't been, we'll look at that later, we can just decompose this tree into this plot right here, which is basically the series of branching events. As you can see on the y-axis is the log scale, because as we get more species, we start accumulating more species, even if the rate's constant. We're accumulating basically geometrically. So we can decompose this really complicated structure into a really simple problem, at least for the problem of estimating diversification rates. So that's kind of mathematically convenient. Now, a thing we discovered in the course of trying to figure out this problem of how to quantify our uncertainty is that this is all the data, all the information in the data is contained in this kind of branching times of the tree. The tree topologies can be just thrown out. And it turns out that for any speciation extinction model of the data, the likelihood function can be written only in terms of the deterministic series of branching times. So right here we have the observed series of branching times that we have in our tree. And for any speciation extinction model we can come up with in a stochastic process, the likelihood function can always be expressed solely in terms of the deterministic series of branching times. Now, right here I showed you actually one of these kind of things which we call a DLTT, deterministic linear tree time plot. And what you'll notice here is that there's no squiggles, right? It's completely straight. And this is the deterministic linear tree time plot for a constant rate birth death model. So we can take a model we have. We're going to assume, say, for instance, constant rates. We're going to assume rates are variable through time by some function. We're going to say they vary across the tree by some means, whatever we want. We can always decompose that idea into a simple deterministic function. And that's actually a really powerful thing. And because not only do we not have to know the tree, we don't actually have to know the details of the model. We can just look at the deterministic output. And so basically, our likelihood of our data is going to be the difference between the straight line that we've theoretically derived and the squiggly line we've observed. Basically, the deviation between those two lines. So what does this mean, and why is this useful? So if NT is the true number of lineages at time tau, and remember, we say true number of lineages because we haven't observed much of the process. Much of the process is completely obscured to us by subsequent extinction events. But at any given time in history, there must have been NT, let's say there's NT lineages. So MT is M tau, is the number of lineages in our reconstructed tree. Okay? Again, we've only seen a sample of the process, so we don't see the true N, but we see some fraction of it, which says M of tau is equal to the true N of T times the probability that any given lineage observed at around the time tau was either gone extinct or wasn't sampled in the present. So we've basically lost some information in this process. And what we can do is we can define something called the polled speciation rate, which we're just going to define as the reciprocal of this M curve times the rate at which it's changing. And basically we call this polled speciation rate because if there is no extinction at all, the number of lineages we observed or reconstruct at time tau is the exact same as there was. There's no extinction event at all. We would just see everything. And instead, this speciation rate is pulled downwards by subsequent extinction events. And so what this means here is that because the likelihood of the model, we can write the likelihood function only upon the reconstructed or the deterministic limit of the reconstructed number of taxa. It depends only on this parameter lambda p and nothing else. All the information about the model is encoded in the deterministic lineage through time plot. Therefore, and this is the big conclusion, is not the total conclusion, but is that any two models with the same exact deterministic prediction for the lineage through time plot will have an identical likelihood function. So any two models which satisfy the requirement, they, in the deterministic limit, as the data size of data sets gets large, any two models which satisfy this conclusion will have the same likelihood and will therefore not be distinguishable using kind of likelihood methods. And therefore, any two models which have this property will be considered equally good explanations for a given tree. So in any given scenario, just to be clear, this lambda p may vary, right? As the deterministic lineage through time plot changes, the curvature changes, so does the polled speciation rate. I said here, again, the polled speciation rate is just the regular speciation rate here, underlying it here, times the probability any lineage around that time will be sampled in the process. So just a little trick here. We can then derive another set of variables from this, which basically puts the extinction parameter in here. I won't go into how this is derived, but we have a differential equation here, and we can write this down like this. So we can estimate this parameter instead of just lambda p, we can decompose it into RP, polled diversification rate, which contains our original lambda and mu. That's a nice trick. With initial condition lambda p not. So this is basically an alternate parameterization of the same exact idea. We only have one piece of information, this lambda polled diversification rate through time curve. So we can write the likelihood down in two different ways. We can write it down in terms of this polled speciation rate or in terms of the polled diversification rate in conjunction with this instantaneous rate of the present. So we basically can either have decomposed the entire information of the model into one term or two terms, depending on what you think is most interpretable. But here's where it gets nasty. And the reason it gets nasty is because we naturally want to know, we have some process we've developed, and this process produces a deterministic lineage through time plot, which we can mathematically derive. And normally we'd be pretty excited about this because then we can take our data set, our phylogenetic tree we've observed, and go back and estimate the parameters of our stochastic model. And we can test hypotheses about what may have caused these variation in speciation rates and extinction rates through time. And that's awesome. But the problem, if you think about it in a weird way, it becomes you also want to know something else. And this is, are there other potential explanations which might be equally good explanations for my data, but are different? So I'll say it again. You have some stochastic model. You can write down a likelihood function for it. You can build your phylogeny from genetic sequences, and then you can say, given the topology of this phylogeny, what are the best estimates of these parameters in my stochastic model? And that's what most people do, or everyone has done in this field until now, including myself. But the second thing is there alternative formulations of the model that are qualitatively different, but which produce the exact same expected distribution of trees? In other words, is there perhaps non-identifiable processes, and can we find them? And this little mathematical trick we have actually says we can find them. In fact, it's really easy to find them. Because if you can formulate your entire problem in terms of this space, this RP, all you can do is you just basically take a mu, take your extinction rate, change it to something else, and then solve for the corresponding lambda. And you can basically, so you can do this, solve these kind of differential equations. This is a nice formula. So you can basically change your extinction rate. Correspondingly, by this formula, change your speciation function in an opposite way, basically. And you can find two models which have two different curves for speciation rate and extinction rate. So such right here, on the left-hand side, we have our original model, which we derived and we fit. On the second-hand model, we have this alternative model, which has a different speciation function, a different extinction function, but by our definition, because all the information is contained here, has identical likelihoods. So the proof is basically in our paper we have is that this works. You can take any given model you have and find alternative models that produce the same likelihood. And now you may want to say, well, how many models are there in practice to have the exact same likelihood? How many models I should be considering? And the answer is infinite number. Because you can basically, mathematically, it turns out, just take your mu parameter that you have in your model, let's say you assume it's constant, change it to be an exponential function, change the lambda in the corresponding way, and have an identical model. And this is really, really, really bad. And the reason it's really, really, really bad is because it suggests that what we're doing has been completely unidentifiable. So if we structure right here, we have 4 different models. Right here, we have the speciation rates for these 4 different models. These have 4 different dynamics. One goes down here, loops up. One's straight. One's got a bump here and goes down here. And these are 4 very different, quantitatively and qualitatively different scenarios. They have very different extinction kind of functions. Very different net diversification rate functions, which is basically the difference between speciation and extinction. And they all produce identical likelihoods. We can never distinguish these apart, and we could do this all day and for an infinite amount of time and keep producing new curves which are qualitatively different and have identical likelihoods, which means we can't distinguish them with any data. Yeah?
So is it kind of like introducing an extinction and a speciation rate gives you so much degrees of freedom in the space of models that you just can vary them in almost, well, an infinite number of ways to basically get the end boundary condition?
Exactly, essentially, yeah. Is that you have 2 stochastic things, which it turns out you can vary in really intricate, nice ways to always get to have the identical boundary condition, basically. Yeah. And this problem was not recognized for a long time in thousands of papers. Does this really affect inferences? Yeah. So here's a tree of seed plants. It's huge. It's like 80,000 species. And you can characterize the lineage through time plot and say, oh, it's growing exponentially. But we can fit two alternative models, one in which the rates are increasing through time, and one in which they are decreasing through time. And these have identical likelihoods because they produce the same whole diversification rate, the same net deterministic limit right here. So we can say with equal probability that diversification has increased through the history of angiosperms or decreased. And we can never tell you which is more likely. Now, that's kind of bizarre because there's literally 1,000 papers that do just that, and including many of mine. I haven't written thousands of them, but I've written many more than I care to imagine. So a great example here is from whales, charismatic group. Everyone loves whales. There's this really nice study that had nothing particularly bad about it, but this group took a whale phylogeny and then wanted to try and explain changes in whale diversity through global temperatures because of whatever hypotheses, doesn't matter, but how efficient they are as feeders, et cetera, et cetera. And so they fit a model in which speciation rate varied as a function of historical temperature using historical data. And so what we did is we took their model, and here's on the model on the left is their data. Okay, we're good. This model here in blue is the model they fit. They assumed a constant extinction function and then fit a curve where the speciation followed quite nicely the global temperatures. We gave an alternate parameterization, and instead of making extinction constant, we just made the difference between speciation and extinction constant. And all of a sudden, we got dramatically different historical curves, which were unlinked to temperature completely. So that their apparent linkage to temperature in the past was completely a consequence of making a quite arbitrary decision about how to set the functional form of the extinction parameter. Okay. And so we have this kind of a conceptual diagram which may be useful to you. Some of you, I'm not that much of a visual person, but it might work. Is what we can consider as things called congruence classes. And we call these congruence classes are any models which have the identical likelihoods. Right? So even if we had, for instance, the true model generating process, and we had, this is the actual thing which generated the data. Maybe speciation extinction rates varied through time in this such and such a way, or varied through temperature, and we had that exact process. There's a bunch of other processes which all have the same likelihood. They're all very different. Now, we're going to consider two cases. The first is that none of the models in our class of models we're considering matches anything in the congruence class, or isn't that the truth? Obviously, we're just approximating the truth. But that basically, we're going to pick the model in our set of models that's closest to a model in the congruence class. So it may not be even the model, it's not the model closest in parameter space to the true model, it's the model closest in parameter space to the model which is like our model we fit of the true learning process. Oops. And the second case is even when we have if we have a model that actually lies in our congruence class of the set of models that's equally likely, we're going to pick that as our favorite model, even though there's another model in our candidate set which is actually closer by some geometric definition in the parameter space to the truth. So there's no reason to suggest that picking a model in our set is going to be closest to the truth at all. In fact, it's basically a random number generator as far as I'm concerned. And so that means that for any model that attempts to estimate lambda, mu, or the difference between them, lambda minus mu, which includes every method to date, there is an infinite set of alternative models that can explain the data equally well. So every one of these functions that I talked about, and there are dozens of different packages, software packages which fit these models, and thousands of papers which they've been fit to, they all estimate these things, which it turns out they can't estimate reliably. And so the take home is we've been extracting more information from phylogenies than is actually available. That's pretty terrifying. It's been actually throwing me into a somewhat of an existential crisis because I have tons of work in which I no longer can stand behind. But that's okay. That's what we learn about life. Now I'm currently, so we solved this problem completely for the extant case, and now we're looking for the case which is the case in, say, viral phylodynamics models where you have sequential sampling through time throughout the course of an epidemic, and we're trying to apply the same reasoning to solve this problem. It's mathematically quite similar, but different. This is what I'm working on right now. And I'm also now trying to basically figure out how reliable, or can we quantify how reliable our inferences are from, say, studies of viral epidemics. And are they equally sensitive to pretty arbitrary choices about functional forms? So that was a bit of a downer, at least for me. Maybe it's interesting to you. So what, if anything, can we say? First thing, this is one thing I kind of mentioned briefly, is that we can actually estimate speciation or splitting rate quite well at the present time, and that's because we have no extinction events to any lineage that's alive today. They're obviously not extinct by the very nature of us sampling them. So if we're only interested in the very recent split events, then we may have some kind of idea of maybe accurately estimate them. So this is from a paper that my colleague Dolph Sluter and I wrote 2 years ago. And basically, there's mammals here, and these are sister pairs of basically recent splits. And what we showed was that the species, or we didn't show, Weir and Sluter showed, we just reinterpreted the data, that species that were living in the high Arctics were actually more recently diverged than species pairs living in the tropics. And this might suggest that speciation rate has been faster in the temperate regions versus the tropics, at least recently. Kind of a cool finding. So if we're only interested in the very recent stuff, I think we can do pretty well. If we're looking at historical changes, we're in shadier ground. And so what's the lesson for you who probably didn't care that much about diversification models before you walked in here and probably won't after you walked in here? Basically, a lesson that you probably already know if you guys are already into machine learning is there's kind of two approaches in statistics. And I borrowed this terminology from Bremen, a famous paper that I'm sure many of you have read. My entire training and my entire life has been about basically developing stochastic models and then basically deriving them in nice ways and then finding data sets and developing kind of computational tools to fit those models and recover the parameters. And so basically what we're doing is we're taking some kind of description of the world. We're imagining that the world works like this, that nature runs its course over time, and at the end we get a tree shape, and we can then, our tool as inferential scientists is to go backwards and reconstruct that history. And if you assume that you can know the data generating model, whatever it is, maybe it's a constant rate model, maybe it's an exponential variable model, that you can infer the parameters of the model. And this is my entire training in science and the way I was brought up and the way I was taught to believe by good people. But there's kind of an alternative way of thinking about science, and I think this is reflected in the machine learning world that's taken off and become so powerful, is basically you imagine that, okay, there's some process that happens in nature, and we don't really know what it is. And nature does its thing, and in a day we get a tree shape. We look at the tree we've sampled, that's awesome. But instead of trying to go backwards and infer the actual parameters of the generating process, we're going to find nice probability distributions which describe the data pretty well. And these probability distributions, let's just call them, I don't know what this curve is, maybe it's psi something, I can't remember what it is. Don't speak Greek. And, but this probability distribution may be consistent with many, many different types of generating processes. So rather than looking for the exact parameters of the generating processes which we assume, we're going to look for nice distributions which kind of capture the general features of our data, even though these may be consistent with multiple different hypotheses of how nature works. In other words, we're looking for classes of explanations. And I kind of hinted at this above, but there's this kind of nice function, fold diversification rate. And it's neither the lambda curve nor the mu curve, but it's some aggregate of the two times by the proportional change in the speciation process. And we can actually estimate this using maximal likelihood or Bayesian inference from the data directly. And so what I've done here is we have all these empirically derived linear street time plots, and I've just fit a nice distribution through these data, which is it works pretty well with very few parameters. And I can use this to derive the estimate of this kind of fold speciation or fold diversification rate. And so we can capture our data very well using very few parameters, which is nice. And, okay, off skip. So then we get right here, I'll just skip this. So then we get a kind of a function, this fold diversification rate through time for all these different groups. And perhaps we can try and learn something from this. This is a huge bottleneck, as you can imagine. We no longer have a single model, but we have classes of explanations, and we're trying to figure out how to make sense of this and how to even talk about this. So I'll go back. We're still working to figure out what it means. We know now we can describe the data very well using our methods, but we don't know how to make biological sense of it. In other words, we have an extremely accurate prediction machinery, but extremely poor explanatory machinery. So at least for somewhere, and we know what we're doing is robust. And so, yeah, that's kind of where I'm going to end my talk today. If you took anything from it, from my struggle and from my learning, I think it's worth thinking about, especially those of you who are working with stochastic models that describe nature in some way. It's oftentimes where we stop, and where I've always stopped is basically saying how well does one model explain data versus some other model, or what are the parameters of the model I'm interested in. But there's a kind of a deeper question, which is, are there alternative kind of explanations that explain the data equally well that I haven't really considered? And is there a kind of a way to go and find those explanations and figure out how broad the possible space of explanation ranges there are? Yeah, this talk was only like one recent project that I did. Broadly interested in kind of phylogenetic models and phylogenetic statistics, in macroevolution, in viruses, and now increasingly in immunology. And I'm an advisor in the Bioinformatics program, so if you want to talk to me, here's my email. And that's it. Any questions? Yeah.
So, yeah, that's really kind of intriguing to me. I admit this is a naive comment, a question,
yep.
But I'm not in the field. But it's actually surprising that this hasn't been thought of before. In sort of naively, the language I would have thought to describe this as that the process of tree is a underdetermined problem, I guess.
Yes.
And you have too much freedom in describing extinction and speciation. So I guess in this question, the virology, epidemiology problem, where you've got all this detailed information, almost seems to be an overdetermined problem, and the simple speciation and extinction rates would not be sufficient to capture the data. And do you have to then include other assumptions, such as variable extinction across the tree?
So I think in the case of viruses, so I'll answer that question in two phases. The first one is why it's not noticed before. These things are actually tricky to find. so I think, one, we should have actually looked at this more closely before. I think that as a field, I think there's lots of sociological reasons why we didn't. But in practice, it's like these models work really well, because if you assume the model and you stimulate into the model, you can recover parameters very well. And you can also, so that gives you a lot of confidence that you can kind of compare models and be like, okay, well, our model comparison works if we compare two models. But the trick is that you only happen to run into this problem if you sampled two models in your model set which satisfied exactly this equation, which is probably not likely. If you just, of the possible models that you concocted in your brain, in your series of models you compared, it's very unlikely, and we kind of do some mathematical reasoning about why this is the case, is like that you would happen to land exactly on these functional forms which are like specified by this equation. So people have done plenty of simulations, plenty of model comparisons, but in the limit they all seem differentiatable, even though people for a long time were quite uncomfortable with that fact. I think most processes, both in the tree of extant organisms and in virus trees, all show heterogeneity in the process across the lineages and across time. And this is definitely something we should take into account, and people do take it into account. They do fit these models. With regard to the exact conclusions we can draw from the viral models, I'm going to save that for the pizza and beverage time because it's quite mathematically complex, and right now it's still somewhat speculative, so I don't want to be on the record explaining it. Yeah. Yeah.
So also a bit of a naive question. Do you think that like going back to really orthogonal data like fossil record will be able to constrain what models are correct?
Yeah, so for sure. So this is something that we like think is great, that like for groups which you have a good fossil record, you can kind of use these two things in kind of together, and people are doing this for sure. I'm not like first person to suggest this. There's like kind of a renaissance in using fossils in combination with phylogenies. And that's something we should develop for sure, because it's a different data set that might give us different information. But it also is the case that for many groups, especially those we care about, like bacterial diversification or plant diversification, or we have very little in the realm of fossil record that's reliable.
And kind of related to that, if you were to take two different phylogenies and look for a concordance between them, would they kind of be orthogonal? Like if you did archaea and bacteria,
so
look where they overlap?
this is the sneaky detail, is that oftentimes when we're say like we wouldn't, for biological reasons, the whole part of this theory is like this realm of theories is we wouldn't necessarily expect the process to be the same in archaea and bacteria. We may expect like bacteria are splitting niches differently or whatever. I don't have any solid evidence or explanation, but I'm saying a lot of people would say, well, archaea and bacteria are very different. We should expect very different dynamics. So they're like the fact that they're in concordance is perhaps consistent with the idea that different dynamics have gone on. But you can't basically use one to like inform the other if you believe that everything's different. You see what I mean?
Kind of. I'm just wondering, like, you know, at the level of analysis, like, use. So I see what you're saying. It is a bit of a circular argument because you're saying, oh, if something's different because it's on a different branch, then you know any differences could be just because they're evolving differently. But at the same time, if you think that there's macro, like say if you were to take temperature, right, like global Earth temperatures, you would kind of expect similar trends between something like archaea and bacteria over these long time periods.
I think that's like a super good insight that I'm like trying to wrestle with right now as myself, is like, to what extent can we port, if we can basically have replicate phylogenies, if you imagine that bacteria is a replicate of archaea, or that different families of plants are like replicates of each other tracing some global temperature, and they should have similar kind of structure to the data, then I think we can get a lot of headway into that because of that. But yeah, I think that's up to debate for sure. And that's something I'm trying to wrestle with.
Like trees of trees.
Yeah, trees of trees, exactly. Any other questions? Yeah.
I think I just wanted to like clarify in my head. So when you're saying that these two models are congruent.
Mm-hmm.
So if you take presented a sample of your data, there's still congruence.
Yes.
Right?
Yeah.
So you can't like solve it by like trying like
No.
a subset of the data.
For any subset of the data, so it's basically the congruency is a property of the models, not the data. Right? So, and there's nothing more in the data that you can inform besides a series of branching times. So you can't like, oh, I'm going to add topology or add like like the tree shape or something other like that. So it's like all the information in the likelihood is defined by the series of branching times, and the congruency is defined based on the model. So any data set you fit, if two models are congruent for one data set, they're congruent for another data set.
That's scary.
Yeah.
I was wondering if you could comment on whether labeled trees sort of cancel the notion that the entire tree can be reduced down to the lineage through time plot. Like ancestral relationships.
So that's a tricky question. And so I'll just repeat the question. Is basically what Angela is saying is that this decomposition of the tree structure into a series of branching times is only a complete representation of the data if we assume that all branches are exchangeable, that we don't have a label associated with them. And that's because basically, even if this tree was swapped, I mean, so that this node was over here, and this node was over here, or any kind of swapping of these things, it would produce the exact same branching structure, number of lineages through time. If you have labeled trees, our proof doesn't apply, but I think there is a version of it for that labeled trees, which has been so far hard to prove, but I think it's out there. Any other questions? Sure.
This is my field. It's another naive question, I think. But do you have a sense of, well, first, like, has anyone sort of tried it this problem? Or, like, got a sense of it and talked about it a little bit, didn't go through?
Yes. So there's basically, since the early 90s, there's been lots of rumblings about this, like, oh, are they sure that you can tell them apart? And there's been, I think, very special cases where people have showed a number of cases where, like, oh, these two models you can't tell apart. And so that was definitely produced, and people were kind of like wondering for a long time, is there a general approach to this problem? But I think people just got stuck. So ours is basically like a generalization of every finding before us. So yeah. So everything you can basically show every result that shows like two models are the same is like just basically it can be a subset of our results, or basically deduced from our results.
And do you sense, well, I don't know, some of it's preliminary, do you sense change in the uptake of this idea in the field, or among people who use this kind of inference with their trees?
I hope so. Well, I've definitely changed my reviewing practices, and I hope other people do. We're trying to publish. I think we will publish in a very prominent place, so that hopefully people will see it. Right now it's on bioRxiv if you want to check it out, Luca and Pennell, and hopefully it's coming to a journal near you, and people will see it and hopefully take it seriously.
Okay, let's thank Matt again.